Okay, really happy to introduce Brian Sedio. Brian is a University of Michigan EEB graduate. He came in 2007. He's actually my first student to graduate. Not my first student, but very, very proud of him for making it through. He graduated in 2013. So some things that stick out about Brian's application. He had a dual major. He majored in forestry and he majored in chemistry. And when he was an undergraduate, every summer he managed to go and work at a different place in the tropics. He worked in Guatemala, he picked up Spanish, and then like Marjorie and Abby, he worked with Lissy Coley and Tom Cursor, doing plant-insect interactions in Manaus, Brazil. And so when he came to the EEB program, he was interested in combining forestry, chemical ecology, continuing what he learned with plant-insect interactions, and then he took on this new dimension of being interested in coexistence of tree species in tropical forests. And I've tried to follow what Brian's been doing. He's kind of a powerhouse in gathering data. After leaving Michigan, he worked at STRI, Smithsonian Tropical Research Institute, for several years, and now he's an assistant professor at University of Texas, Austin. So let's give Brian a warm welcome back.
It's really a pleasure to be back here. So thank you so much for inviting me to be back. It's been 11 years, and I really love this place. So it's special to be here. Okay, so I'll get started. What's always motivated me is the big question, and that is, what determines species diversity? And any attempt to answer this question involves the latitudinal diversity gradient and the incredible diversity of tropical forests. So to put that in perspective, in global temperate forests, there are just under 1,200 tree species in 4.2 million square kilometers. And yet there are 3 forest plots in the Amazon that are a quarter of a square kilometer, and they each contain more tree species than that. Now, to put that another way, within the forest geo network of forest plots, a forest plot in the boreal forest of northern Canada contains 11 tree species. A typical forest in North America, actually a quite species-rich forest in North America, contains 65 species. And the most diverse plot near Manaus, Brazil, contains 1,400 species in the same area. So there's also a dimension to this diversity that often goes underappreciated, and that is that a handful of really diverse tree genera represent a large part of the diversity of most tropical forests. So botanist Al Gentry referred to these as species swarms for their incredible local diversity and the often apparent physiological and morphological similarity within these genera. And so in Barro Colorado Island, Panama, where I do much of my work, the 5 most species-rich genera represent almost a quarter of the tree species on the island. And so these tree genera, they challenge our understanding of coexistence, because in order to stably coexist, species need to be doing something different to exploit different resources or otherwise minimize their competition. But these lineages also present an opportunity, because if we can put everything in a phylogenetic context, we can understand what drives the diversification within these lineages and what distinguishes species and what might facilitate their coexistence. And so foundational hypotheses in ecology and evolution posit a central role for plant-enemy interactions in both the maintenance and the origins of the diversity in plant lineages. And yet what's interesting is if you actually look at herbivore or pathogen host use patterns, they're often not as specialized as Jansen and Connell would have presumed in their model. And what you see is phylogenetic signal. So you see beetles or caterpillars or pathogens that attack many species in a genus in a forest. And yet, on the other hand, these species-swarm tree genera are famous for their chemical diversity. And just to give you an example, Psychotria, which I focused my dissertation research on here at Michigan, they're famous for their psychotropic or hallucinogenic alkaloids, such as the active component dimethyltryptamine in the famous hallucinogenic tea known as ayahuasca in the Amazon. And so I like to, there's a Peruvian shaman named Pablo Amaringo who paints his visions when he consumes ayahuasca. And they're really interesting to look at. And I like to use these as a sort of a illustration of the chemical diversity or the pharmacopoeia of chemical diversity of the tropical forest. And so, I started grad school and I was thinking about all these ideas as I hiked around in the jungles in BCI. And I noticed that in some places, I would see two different species of Psychotria that were clearly getting attacked by the same chrysomelid beetles. And then I would notice in other places, it was clear that the herbivores that were destroying this individual on the left did not view the species on the right as a viable host plant. And sometimes people express skepticism that these interactions really matter. But I visited this same site the following year in 2010, and it mattered for that Psychotria. So I think these definitely affect fitness, I would argue. So the big questions that motivate my work, which were originally inspired by my observations of the Psychotria in BCI Panama, are do the chemical differences among even closely related tree species in these species swarms represent niche differences that might facilitate the maintenance of diversity, allow them to coexist by minimizing their competition? And secondly, is there evidence that this is a mechanism of diversification that might explain the evolutionary origins of diversification in these lineages to begin with? And then finally, is there evidence that variation with latitude and climate in these mechanisms might explain the really dramatic diversity gradients that we see at the global scale? So, as we've seen, the hypotheses that were posited to answer these questions, some of them are 50, 60 years old. But in much of that time, we've lacked the tools to study chemistry on a scale commensurate with these questions. And that's all changing really rapidly. So the folks on this slide have all contributed heavily to an explosion in metabolomics. And it's all driven by bioinformatics. It's not really how we collect the data. It's what we can do with the data. And so, I'm not a computer scientist. I've really just been an early adopter of everything that these folks generate. And so in the rest of today's talk, I will explain how this bioinformatics of metabolomics works. And then we'll go through a series of vignettes. We'll look at forest chemical ecology. We'll ask this question of whether chemistry represents niches that sustain diversity. And then we'll look at the potential role of chemical defenses in driving diversification and adaptive radiations. So, like I said, the different ways of collecting the data, the most important thing is to have a really high resolution mass spectrometer. Usually those are coupled with liquid chromatography systems. And the best, QTOF or an Orbitrap system can do it, but the key is to be able to resolve masses at a really fine level. And so when you collect mass spec data, the x-axis here is the mass to charge ratio. The y-axis is just ion intensity, the number of ions that are hitting the detector. But a lot of the techniques we use rely on tandem mass spectrometry, which means you blast the molecule to pieces and you measure the mass to charge ratio of all the pieces. And that's really key because molecules with really similar structure will break into many of the same pieces. And so you can compare that fragmentation profile to compare the structures of unknown metabolites. And so the first big innovation in metabolomics that allowed us to do much of what we do is called molecular networking. And at its core, it's a pretty simple mechanism. So let's imagine we have these 7 tandem mass spectra. We've smashed each of these molecules into bits and we're comparing them. So you can treat each one as a vector. And to compare two vectors, you can measure the cosine of the angle between them. And so 2 vectors that are really similar will have a cosine close to 1. If they're completely different, it'll have a cosine of 0. So what we can do is we can measure all these. Essentially what you're doing is comparing the fragment masses. We can compare absolute masses. We can also account for shifts, which is like, let's say one of these is bound up with a potassium ion, but the other one with a sodium ion. So it's really the same thing, but they all differ by exactly the same mass. We can recognize that and tell that they really are the same. And OK, so let's say we have these 7 spectra. And we want to compare everything to everything. And we have these similarity scores, these cosine scores. Well, then what we can do with that information is represent unique spectra as nodes and similarity scores above some threshold as links and turn this into a network. And this seems trivial when I show you, 7 metabolites. But what's powerful about this is it's scalable. So we can scale up to looking at thousands of metabolites. The other thing that's interesting is if you have a library of known metabolites, and we've collected these fragmentation spectra, then we can compare them to these networks in a process that's kind of roughly equivalent to blasting a DNA sequence. But we might not know what everything is in that whole network, but if I know that that little orange molecule is caffeine, then those other molecules that are clustering with it, I know they're structurally related to caffeine. And that might tell me, in a chemical ecology context, most of what I want to know about those compounds. OK, so it's like blasting DNA sequence. So the other thing is we can map features. So we could map the mass of the molecule, or whether it was found in species A or species B or both. And this is what lets us pursue chemical ecology. And then finally, if you zoom in on one of these networks, so here we have the parent mass, the mass of the molecule before it was fragmented. Now, to most people, we would look at this and say it just looks like a bunch of numbers. But the differences in mass can tell you exactly what distinguishes these structures from each other. And that's valuable information. So more recently, there have been a couple developments that have really opened up what we can do with metabolomics. And one of them relies on isotopes. So if you look at a mass spectrum, this is kind of a gross simplification because they don't really look like this. What they look like, you always see these tiny little peaks. They're just really sloppy with all these little peaks all over. And what those are isotopes. So this molecule is caffeine. And so the big peak, these are all the caffeine molecules in which every single carbon atom is a carbon-12. But that smaller peak, these are the smaller proportion of caffeine molecules that contain one carbon-13. So it weighs more, and we can measure that difference on the mass spectrometer. And that smaller peak are the proportion of caffeine molecules that have two carbon-13s or one carbon-14. And now, for reasons of nuclear physics, an extra neutron in a carbon nucleus doesn't have the same mass as an extra neutron in an oxygen nucleus or a nitrogen nucleus or what have you. And so with a very high resolution mass spectrometer, which is what we run, we can tell the differences in these isotopes of different elements, which means we can predict the formula really accurately. And most of what you want to know about a molecule is represented by the formula. Even just the relative ratio of these elements can tell us whether it's a carbohydrate or an alkaloid or an amino acid. But if we have the formula, there are only so many structures that formula could represent. And so if you have a formula and you have a fragmentation pattern, it becomes a matter of probability. What is the most likely structure with this formula that gave us this fragmentation pattern? And so we can use that to predict the structures of unknown metabolites. And what's interesting is there's a separate algorithm that predicts the most likely metabolic pathway of origin. And we don't just use the structure, the predicted structure, to classify it, because we can get a more accurate classification of the metabolic pathway of origin by using the formula and the fragmentation pattern. And so here's an illustration of what we can do with this. So this is a dendrogram. It looks like a phylogeny. This is actually a dendrogram of metabolites. And the branching pattern reflects their structural similarity. This is over 8,000 metabolites that represent all 314 tree species from the 50-hectare forest plot at Barro Colorado Island, Panama. And what I've done here is I've colored the branches to represent the major biosynthetic pathways of origin of these metabolites. The other thing I've used in this figure is I've put a bar plot around the outside that shows how many of the tree species each metabolite was found in. And so you can already start to see patterns. Like you see the red color represents fatty acids. You can see that each fatty acid was found in many or maybe even all of the tree species. Whereas you look at that kind of teal color that represents alkaloids, and each alkaloid is found in one or maybe a few tree species. So tree species are more differentiated with respect to alkaloids than fatty acids. So there's one more step in this process that I'm very excited about that is relatively new. And that is that if you can predict the structure, we can sometimes identify actual known compounds. If you can predict the structure, though, you can use that to calculate chemical and physical properties of the metabolites. And this is also a trait, potentially a functional trait of plants. As I'll show you later, I think it is. And so I really got this idea from an IDIV working group that published this paper before they invited me to join the working group. So I'm not actually an author on it, but it's a cool paper. So what they did is they looked at all these different chemical and physical properties and found Now, a lot of these are really highly correlated. And some of these, it's really obvious, like the molecular weight and the number of atoms and so forth would be correlated. But basically, there are really only 5 axes of variation in the chemical properties of the plant metabolome. And here's a figure that illustrates, in their data, the molecule with the lowest and highest value for one property selected from each of these 5 groups of highly correlated traits. And so you can kind of see this is the space within which plant metabolomes vary. And so, so I think this is exciting because there are different ways, different facets to how we can think about plant metabolome variation. And to give you some examples of chemical and physical properties and their functionality, so aromaticity affects the capacity of a molecule to absorb light, such as, for example, high elevation plants that are exposed to ultraviolet light radiation. They need to have more light absorbing molecules to protect them from UV damage. Another variable of interest is polar surface area. That is negatively correlated with passive membrane transport. So if a molecule is going to slip through a cell membrane to enter a cell, it needs to have a non-polar surface. Another interesting variable is it's called FSP3, or the carbon bond ratio. So this is the ratio of single bonded to double or triple bonded carbons in the atom. Very simple metric. It is pretty strongly correlated with the probability of a molecule making it through pharmaceutical trials to become an approved drug. That's interesting. So we'll call it complexity or bioactivity. Of course, there are other properties. There are quantitative properties that we have to measure, like toxicity. Some of these are context dependent. A molecule might be toxic to one thing and not to another. It might be heavily dependent on concentration as well. 2 other mechanisms by which molecules affect insect herbivores are oxidative capacity and protein precipitative capacity. So things don't have to kill the insect. They can just like gum up all the protein in the insect's stomach so it's not getting anything out of its food. And then finally, as we'll see, a really important property that is relatively unique to plant metabolomes is qualitative, and that is the dissimilarity in composition. So 2 plants might not need to be super toxic as long as they have different compounds, and that might allow them to avoid the same pests and pathogens. So, and then finally, that last thing, to measure the similarity, I came up with a metric to take advantage of these molecular networks in order to account for the structural similarity of distinct molecules when comparing 2 different plants. And so to give you an example, let's imagine that the Psychotria on the left contains caffeine but not theobromine. And the Psychotria on the right contains theobromine but not caffeine. Well, if we were to use Bray-Curtis similarity, the similarity would be 0 because they don't share a compound. And that's really common in nature. I mean, plants, there's so many compounds, very few compounds are shared. And so you get a bunch of zeros if you use Bray-Curtis similarity. Well, in a molecular, so first of all, these only differ by 1 methyl group. So, you know, and are they really even different functionally? I mean, they have the same effect on humans. In a molecular network, these would be really tightly linked with a cosine score of 0.95 or something like that. So what we came up with is we could calculate the structural similarity matrix. So this is, if we have a network of, you know, 1,000 metabolites, this is 1,000 by 1,000. So it's comparing every molecule to every molecule. How structurally similar are they? Then we can calculate the compositional similarity matrix. This is the ion abundance of every molecule in species A and the ion abundance of every molecule in species B. So both these matrices are 1,000 by 1,000. We can multiply them together and take the sum. And what that gives us is the abundance-weighted structural similarity of the metabolites in the 2 species or samples. And again, that's important because this example, where one species has caffeine and the other has theobromine, would have a Bray-Curtis of 0. But if we use my CSCS metric, it has a non-zero similarity because the compounds they have are similar. OK, so when I came up with this, I felt like I had a hammer, and just the world just looked like it was full of nails. And so, oh, what the heck? What just happened here? It went to like, hold on. It went to the end of the talk of like slides that I dump at the very end. I like never delete anything. Yeah, this is almost where I was. OK, great. OK, so anyway, let's move on. So you know, I turned my attention to the forest geonetwork. So this is a global network of forest plots. Scientists all over the world have set up these plots. They There are now 78 plots in 28 countries, 7 million trees, 12,000 tree species. And these plots are measured, actually it's every 5 years, I don't know why I wrote 4 there. And so in these plots, the biggest ones are 50 hectares. Many of them are 25 hectares. That's a huge area. I mean, that's like the size of this university's campus. And in that area, every stem that's 1 centimeter at dBH or bigger is identified, measured, and mapped every 5 years. So it gives us a ton of dynamic data over decades. Basically everything the size of your finger this high off the ground in an area the size of this campus. That's a lot of data, and it lets us track the performance of individuals as a function of their neighborhood and their environment. So when I did my postdoc, I kept it simple. I compared a temperate forest and a tropical forest. And at BCI, I had at first species that represented a quarter, so these 7 genera represent a quarter of the species. And I focused on a subset that represented 90% of the stems in the plot. And similarly, in Maryland, these 4 genera represent a similar proportion of the tree richness. And so I built this molecular network, and there's not a lot you can glean by staring at this, but what it allowed us to do is compare the chemical composition and the similarity between species in these 2 forests. And this had become a big question. So, and this is kind of dated now, this is from 2009, but there's this really nice review paper that asked, is there a latitudinal gradient in the importance of biotic interactions? And for a while, there were a series of papers that claimed to find evidence, again, that didn't support this, that tropical plants aren't any better defended. And it was mostly based, though, on leaf toughness and total phenol concentration. And it got kind of, so these are the papers that failed to define that they didn't find that tropical plants were more defended or had more herbivory. And it got kind of spicy. I mean, there were some papers that said, that people that still believe this hypothesis were dogmatic zombies. So I was a dogmatic zombie, and I kept looking. And what I found was that, well, the species in BCI were much less similar to each other than the species in the temperate forest in Maryland. And this is overall. If you look at, what happened here? I think, sorry, got a missing slide here. Anyway, I also came up with a phylogenetic metric. And so you would expect if traits are conserved, there should be a relationship between log phylogenetic distance and chemical similarity. That's what we see on the left. If traits are really diverging among closely related species, and there's a breakdown of phylogenetic signal, then we would expect no relationship. And when I plotted these, well, for Maryland, there's this clear log linear relationship. Right? So more closely related species are more similar chemically. In Panama, we got a flat line. So this suggests that chemical differences accrue at a roughly constant rate in the temperate forest in Maryland, but are under selection for rapid divergence among close relatives in Panama. And so we also looked within genera, and what we found is that if we take the species swarm genera, the 7 largest genera from BCI, and only compare congeneric species, and then do the same in Maryland and only compare oaks to oaks and hickories to hickories and viburnum to viburnum, congeneric species are much more similar in an eastern deciduous forest than they are in Panama. So these species swarms are really made up of chemically distinct species. And not to beat a dead horse, but I think this is a nice way to visualize these results. So here, every point is a species. All the black points are from Panama. All the gray ones are from Maryland. And so you can see that the temperate forest represents kind of a subset of the chemical space of the Panama plot. If I highlight the large genera from Maryland, you can see that not only are they pretty similar to each other, they're also kind of in the middle of the chemical space, all lumped in there. If we look at the big genera from Panama, though, each of these genera represents a larger chemical space than the entire temperate forest. So this is the top 4, then this is the next 3. Myconia is kind of the odd one out. Myconia is almost more like a temperate tree genus chemically. It's going to be interesting to look at. And then finally, this is just to show that the 7 large genera from BCI represent the bulk of the chemical diversity when we visualize it this way. And that might be interesting to think about as well. So more recently, I moved on to looking at North America. So this is kind of I call this my COVID paper, because my whole project in Panama got shut down. And this was supposed to be a big continental scale comparison. And instead, I was stuck with just the North American stuff. And I thought this paper was going to be kind of boring, and it turned out to be actually pretty interesting. So we did the same thing for North America. Here are the sites. We've got a boreal forest in the Northwest Territories and some western coniferous forests. It's quite a bit of climatic variation, and we've got a temperate rainforest in Washington. We've got the Michigan plot at the East George Reserve. And what I found, so if we just look at metabolite richness, so this is ignoring the structural differences among metabolites. We didn't see a relationship with precipitation, but warmer plots are richer, but it seemed to be driven simply by species richness. However, when we looked at chemical similarity, it wasn't driven simply by species richness, but we saw that trees in warmer and wetter environments, even within North America, are less chemically similar to each other. And so that's interesting. Even in a temperate rainforest in Washington, these processes are at work. So now this is my seg because of Martin Wolf, who's a collaborator of mine. He visited my lab in Texas in 2021, and he really loves fishing. And so he went fishing for alligator gar on the Trinity River, and he caught a big one. But Martin is very interested in elevational gradients. And so with Martin, we started looking at elevational gradients in the Alps, looking at willows. And I kind of owe this idea to Martin. So he noticed that, as we know, in abiotically stressful or resource-poor environments, plants actually invest more in defense. And that's because they can ill afford to lose tissue. So even while specialized herbivory is greater at the bottom of the mountain, abiotic stress increases with elevation. There's colder temperatures, it's usually drier. And plants, so what we expect is that plants might actually invest more quantitatively at the top of the mountain. They might even have greater richness of metabolites. But the processes that generate metabolite diversity and divergence are the coevolutionary feedbacks between plants and their living natural enemies. Abiotic stressors don't co-evolve. If it's cold, but it doesn't co-evolve. But natural enemies co-evolve, so there is no one best metabolite. You can't just escalate and invest ever more in the best thing, because the insect or the pathogen will just co-evolve. And so what it does is it selects for divergence. And so what we expected to find is a shift in the chemical space and a greater investment at the top of the mountain, even while we saw greater divergence or chemical beta diversity at the top of the mountain. And so we looked at this in these European willows. And you can see if you look at the whole metabolome, you don't see much. If you zoom in on salicinoids, which are anti-herbivore defenses, the high elevation willows, which are in blue, represent a smaller chemical space than the low elevation willows in orange. So the bottom of the mountain, the willows are more divergent in their salicinoid chemistry. And then if you look at flavonoids, so flavonoids are interesting. They're anti-herbivore defenses, but they also are highly aromatic, and so they act like sunblock. They absorb ultraviolet radiation. So we see a shift that high elevation willows are they're moving in a different direction. They're expressing a different metabolome in terms of flavonoids. Then we also, now I won't ask you to really stare at this, but it is an interesting way, and some of the students I spoke with today I think would really benefit from using this approach. So this asks not whether the two groups are different, but whether they're trending in different directions if you take phylogeny into account. Which is to say, maybe we have these low elevation willow ancestors, but every time one evolves to exploit high elevation environments, it starts moving in a certain direction chemically. So it hasn't got to its destination yet, but if you compare it to its relatives, and you compare every high elevation willow to its low elevation relatives, they're all going in the same direction. That's the kind of thing we can pick up on with this analysis. Okay, so keep these same ideas in mind. I was working with Martin. We were working on these willows in the Alps in Europe. And then COVID was happening, and I couldn't do anything. And so some colleagues in St. Louis, Missouri, wrote to me, and they said, we're sitting on this treasure trove of samples from Madidi National Park, Bolivia, from this incredible elevational gradient in the tropical Andes. And so I started this collaboration with Jonathan Myers at WashU and Sebastian Teo, who's the director of the Madidi program at the Missouri Botanical Garden. And this is an incredible project. They have a whole ton of 1 hectare plots that span from Amazonian lowlands and lowland tropical dry forests all the way to the tree line of the Andes. It was too diverse for us to handle, so we chose 16 of these plots and analyzed 2,000 samples. And so here the y-axis is chemical dissimilarity, so it's 1 minus my similarity metric. And the x-axis variables here are diversity, that's species diversity in the forest plots, elevation, and climate. So we did a PCA, and on the right side of that climate axis are warmer and wetter forest plots. Each data point is a forest plot. And what we see is that the dissimilarity in chemical composition increases with species diversity, decreases with elevation, and increases with precipitation and temperature. Then we pulled out just secondary metabolites because we were able to predict their chemical pathways of origin. And we found that this pattern is mostly driven by secondary metabolites, and it's climate, not elevation per se, that's driving this pattern. And we didn't see it when we looked at primary metabolites. So it seems to be primarily driven by things that are potentially involved in defense. We also ran these structural equation models. And long story short, what we found is that the best models were those in which the effect of climate on species diversity. So warmer and wetter climates have more tree species. That effect, in the best model, that effect was mediated by chemical dissimilarity. Except when we looked at primary metabolites, the best model was simply that warmer and wetter climates have more tree species. Chemistry doesn't matter. But it mattered when we looked at secondary metabolites. OK, and this is where it gets kind of exciting. So I was inspired by the Walker paper and their use of predicted structures to calculate chemical and physical properties. And so I re-examined the Medidi data. And I did this with a recent UT graduate, Sierra Chadwick. And what we found was that these chemical and physical properties vary pretty dramatically over the elevational gradient at Medidi. So this one's only marginally significant, but we see that higher elevation trees are more aromatic. They have a larger pi system, which means they can absorb light. Topological polar surface area increases with elevation. That means the high elevation trees are more polar. There's another variable, so molecular weight doesn't vary. Alpha polarity is actually a strange way of saying hydrophobicity. Here we see hydrophobicity decreases with elevation. That means the high elevation trees, again, are more polar. They have a more polar metabolome. I think what's going on is that having a more polar metabolome might aid water retention in the leaves in cold and dry environments. Anyone who knows about tree physiology can tell me if I'm wrong about that, but that's my interpretation. And then finally, that last variable, this is the one that we call bioactivity, or basically pharmaceutical potential. And notice that it decreases with elevation. It increases in warm and wet environments, which is to say the plants at the bottom of the mountain in the warmest and wettest forests have greater pharmaceutical potential or bioactivity in their metabolomes. So then I started to get creative, and I thought, now bear with me. I'm trying to work on how to explain this in a manuscript, actually, and it takes so many words to explain. But OK, so what I did, if I lined up all the metabolites for one of these variables, and then I took the upper or lower quartile. OK, so let's say it's aromaticity. I took the most aromatic or the least aromatic metabolites, and then I calculated my CSCX similarity metric. So I'm asking how similar are the co-occurring tree species for the most aromatic or the least aromatic metabolites. And so what you see is that when I looked at the upper quartile for aromaticity and topological polar surface area, the higher elevation trees are more similar to each other with respect to the most aromatic and light absorbing molecules and the most polar metabolites. So not only are they more polar and more aromatic, they're converging on the same or structurally similar metabolites with those functions. And then meanwhile, when you look at this bioactivity variable, it also increases with elevation. Similarity with respect to it increases with elevation. But I think that might be more driven by the fact that high elevation trees are more aromatic. But this is consistent with the notion that low elevation trees are potentially more bioactive. So we're writing that up for submission to PNAS. We'll see how it goes. So anyway, this is not the end of the talk, but a temporary conclusion. So with respect to elevation, so again, high elevation trees in colder and drier communities, they're more aromatic, more light absorbing, more polar metabolomes, and they're converging on the most polar and aromatic metabolomes. Low elevation, warm and wet communities, they're less aromatic, less polar, potentially more bioactive. But their key feature is that they're more divergent overall when you take all secondary metabolites into account. OK, so now I have another angle here. So we can do a lot with metabolomics. It really is scalable. So we can collect data at a really large scale. But when I have graduate students and postdocs working with me, it's nice to zoom in on some more manageable project that sheds lights on function. And so often that involves either collecting data on some interacting organism, insect herbivores or pathogens, or using experimental assays to test for the functionality of these differences that we can observe in the metabolome. So I led a team in Panama. We collected 10,000 insects at BCI and then 2,000 in Maryland. And we used backpacks that are made for tailgate parties, so we could collect the insects on ice and DNA sequence their stomachs. And so the idea is, if we know which insects eat which plants and we know the chemistry of the plants, we can model what chemical differences drive these host use differences. And I have 2 postdocs and a student working on aspects of that. Now, here are some preliminary results based on psychotria. So Guillaume Dury is a postdoc in my lab who's working on this. And then another postdoc in my lab, Dale Forrester, is using fungal pathogen bioassays. So basically, we do an extraction of a leaf, experimentally inoculate a fungal colony to see whether it's toxic to that fungus. And so he's doing this for 120 tree species along the Isthmus of Panama to look for variation in toxicity with the environment. He's also looking at insects. It's a little harder. We're not actually doing it that way, but that was a nice picture. My grad student, Fiona McNeil, is looking at nectar metabolomes and the way they drive pollination syndromes in Passiflora and Salvia. And my student, Sarluis Mihango, is looking at allelopathic metabolites. And it may just be that the rainforest is full of plants that are all trying to poison each other. And so we're doing this in collaboration with folks, all these assays actually, in collaboration with Tatiana Lobo at the Universidad Nacional de Colombia and Christopher Boya in Panama. And so our results can then be used by people that do drug discovery in Panama and Colombia and use it to guide their own drug discovery research. So the other element to this is if we know which pathogens are toxic to, or which plants are toxic to which pathogens and insects, we can do this with the whole metabolome, and that tells us something. But what's exciting is that it also lends itself to fractionation. So we can use the methods of natural products chemistry to break the metabolome into pieces based on, in this case, on how hydrophobic or aqueous it is. Or we can use these new devices that can use a column to either break things into fractions or isolate a single metabolite in sufficient quantity to use it in a bioassay. And then ultimately, we can discover a new compound using NMR spectroscopy. OK, so the other aspect of chemical niches, this is a new project. So in Panama, we've got this project funded by Corteva Agrisciences that is funding us to collect metabolome data on 2,000 tropical tree species. We've had a team in the field since 2018. This was the original team. It's kind of grown since then. We've had a lot of interns working with us. And so we march around the forests of Panama with a big liquid nitrogen tank on our backs and collect leaves. Our goal is to sample every tree species in a network of plots set up by Rick Condit that represents nearly 2,000 tree species and a lot of environmental soil phosphorus and rainfall variation. And so basically, we'll be able to look at how the importance of chemical neighborhoods and defenses vary with environment at this regional scale. And my former postdoc, Guille Peguero, worked on this before he went to Barcelona. He's still kind of working on it. And Dale Forrester is leading this and has been leading the efforts on the ground in Panama for the last year and a half. And more generally, this is a group of postdocs on the top and graduate students on the bottom that are working on aspects of this broader project. But I want to share with you one key exciting result that is unpublished. You guys are the first to see it, so this is a privileged audience. So I've already shown you this. This is a dendrogram. Well, my goal for doing this was to be able to say, can we model seedling survival as a function of chemical neighborhoods? So let's imagine on the left, these two seedlings they're chemically different enough that they don't share the same herbivores and pathogens. So they successfully recruited. Whereas these other ones, they're not identical, they're not the same species, but they're too similar. They got hit by the same enemy and they died. So this is the kind of thing we can detect. And my former postdoc advisor, Joe Wright, has been tracking this for 20, actually I think it's close to 30 years now. So he has 184,000 seedling observations over several decades. And there are over 50,000 first-year survival events. So statistically, it's easier to look at first-year survival because it doesn't get complicated, multiple generations. So we just ask for every seedling, we saw it once, did we see it again? Did it make it to the next year? And what we find is this. So if we start at the bottom here, so large conspecific neighbors, anything to the left of 0 means it has a negative effect on survival. So being near a conspecific adult, that's bad. That's the Jansen-Connell effect. We don't see many strong effects of being chemically similar to heterospecific adults. But when we look at the seedling level effects, so here's similarity. In this case, this is density of conspecific neighbors that are also seedlings. So that is even stronger than the effect of conspecific adults. That is this. So being near conspecific seedlings results in lower survivorship. That's Jansen-Connell. But when we look at chemical similarity to primary metabolites, so if we compare each seedling to heterospecific seedlings in terms of primary metabolites, being similar actually has a positive effect on survival. Being similar with respect to secondary metabolites has a negative effect on survival, and it's more than half as strong as the effect of being near conspecifics. And so what I think is going on here, this is my interpretation, if you're chemically similar with respect to primary metabolites, that probably reflects similar physiology. This is a habitat effect. And if you're a first-year seedling, you're not really competing for resources yet. So if you're in a good habitat and there's light and water and you're with physiologically similar heterospecific seedlings, you're more likely to survive. But if you're chemically similar to neighboring seedlings with respect to defenses, you're more likely to share and hence compete with this is enemy-mediated competition driven by insect herbivores and pathogens. And then the other exciting result here is that I saw that result and I wanted to say, okay, well, how many of the species are sensitive to this? And so I looked at the species level coefficients, and I'm familiar enough with the tree species of Panama that I saw the species that didn't have a strong effect, and I went, wait a minute, are those all pioneers? And so I don't know if you guys are familiar with this paper. There's a paper in 2018 and then 2020 by Nadia Ruger that looked at life history strategies along two major axes. So trees can either grow fast or survive, but they can't do both. Like the fast growers have higher mortality rates. It's a fundamental axis of variation in tropical forests, maybe even forests globally. And so what I did is these authors have shown that in these large forest dynamic models, you can get away with basically treating species as one of 5 major categories in terms of these life history strategies. So what I did is I plotted the species level coefficients against the species positions on Nadia Ruger's axis 1. And I found a pretty strong relationship. And what we see here, as you see on my little cartoon on the right, is that the slow growers with generally low mortality that can recruit in the shade are more strongly impacted by the negative effects of sharing secondary metabolites with their neighbors. And the pioneers, the fast growers, are less affected. I also looked at similarity with respect to primary metabolites. And now what's different here is that a lot of these points are above 0, which means the fast-growing pioneers are more strongly positively affected by being chemically similar with respect to primary metabolites. So that means pioneers have a larger benefit of being in the right habitat, being physiologically similar with their neighbors. That makes sense. They need to recruit in a gap. And so I think the sensitivity to these effects might be fundamentally connected to species positions on these basic life history axes. So these are the conclusions. I think I'm not going to repeat this. I'm going to delve into the chemical evolution stuff in more detail at a talk I'm giving tomorrow at the herbarium. So I'm going to skip that right now. So you'll have to come to my other talk. And what I am going to do is quickly show you what I'm doing now. So I have an NSF grant with Annette Osling at UT, who used to be faculty here, and Jonathan Myers at WashU. And we have 2 postdocs, Dale and Daniel Smith, and some collaborators at other places. So we wrote the proposal around these forest plots in the Western Hemisphere. And we proposed to look at climatic and elevational gradients in the importance of chemical interactions for forest dynamics. But I also have been able to scrape together funding to build collaborations with forest geoplots in other parts of the world. And so as of now, it looks like we're going to have metabolomes of about 8,000 tree species we're approaching 60 forest plots worldwide. And so we'll be able to examine all of these, how chemistry itself varies, its properties, its composition, its diversity, with climate and the environment at the global scale. And then because we focused all these leaf collections in the forest plots, we can link it back to the performance of individual trees and forest dynamics, and hence diversity maintenance and the mechanism of selection. So this has been a result of a lot of individual collaborations. These are folks from the Czech Republic that work in Papua New Guinea, some of the European PIs, folks that work at these African plots, work in Southeast Asia, including Thailand and Malaysia and India. And these have been my main collaborators in the neotropics. And I've also been collaborating with some folks at Xishuangbanna Tropical Botanical Garden who have sampled 11 of the Chinese forest plots. So this I've been working on for a long time. It's not finished yet, but I'm pretty excited about where this is going. And then I've used this as I've built these collaborations, I've used capacity building and the training of students in metabolomics methods as my main, that's how I get people excited to work with me on this stuff. So I've led workshops at Xishuangbanna Tropical Botanical Garden and STRI. We led a workshop in English and one in Spanish, which we flew in students from Ecuador and Colombia. And at UT, I teach a graduate course in which I teach these metabolomics methods. And so this has allowed me to I'm like a traveling metabolomics salesman. And I just, preaching the word here. And so in Texas, we've also been able to set up a really cool gradient. So we have a growing number of field stations in Texas, and we have sampled plants at all of these. And obviously, Texas represents some really interesting climatic and biogeographic variation. And here are the ecoregions in Texas, from the Chihuahuan Desert to the Louisiana Bayou. And we'll have this, and even down on the US-Mexico border and subtropical forest along the Rio Grande. And to put this in perspective, so here I've put all the plots, or most of the plots, on axes of mean annual temperature and mean annual precipitation. You can see that the Texas field stations actually represent a pretty nice spread with respect to climate. So I want to say thank you very much for inviting me back here, and go blue.
Thank you, Brian. I thought you did a good job of selling metabolomics. Do we have any questions?
Well, Brian, so for that elevational gradient, and at a higher elevation you see more aromatic metabolite, I wonder how big is the difference of the elevation? If it is big, you may want to consider the atmospheric oxygen concentration. Because I think the whole redox homeostasis balance would come into play.
They are antioxidants, yeah. So that's good. Thanks for pointing that out. It is a very dramatic elevational gradient from the lowlands of Bolivia to the tree line of the Andes. So.
Thanks, Brian. That was super cool. I also have a question about the elevational gradient stuff and the converging on those compounds that help deal with abiotic stress. And I was wondering if you've looked at any of your data across latitude, or if you expect in the forest geoplots, when you look at that, for the higher latitude, more temperate species to also converge similarly on compounds that deal with abiotic stressors.
That'll be a really interesting question that we can examine with the global data set. But I expect there will be differences in properties. And so because our global data set represents variation in precipitation as well, we have some really arid savanna ecosystems. We can look at whether arid environments are selecting for polar metabolomes, and what's going on at really high latitudes, where high latitudes are similar but different than high elevations. So like this issue of oxygen concentration or increased irradiance might be a little bit different. So that'll be interesting. We got a question in the back over here.
Awesome talk. I was just wondering, like, how, of your metabolome, how many actual high confidence structural chemical formulas can you get? Because, yeah, I'm looking at metabolomics data, and it's very hard to get that kind of prediction.
Yeah, and the data collection matters. Like I put a lot of time and effort into optimizing my methods, and sometimes like people will ask for my help to work with like data sets that they generated on their own, and I'm like, oh man, it can be hard. But in general though, so the most confident prediction we have is the formula. The formula, we have very high confidence in that. The second most confident thing we can predict is the metabolic pathway of origin. The formula, or the structure itself, is the least confident. But the folks that developed Sirius tell me that the correct structure is usually in the top 10 of the predicted structures, and we do have confidence scores for those. So you can kind of, if you want to take advantage of that, you could say, I'm going to look at all the top 10 structural predictions and weight things by their relative confidence and account for that variation.
Brian, over here,
Hey,
nice to
Catherine.
see you again.
You too.
A wonderful talk, fascinating. I'm curious about what is driving these, the selection for these chemical differences, and how much you have insight about that, you individually, you collectively, all of you interested in this, and whether a lot of it is being driven by insects or arthropods, or whether microbes